Welcome back to the Whiteboard Biologist. Today, we are going to discuss about RNA, or ribonucleic acid. In this video, we will explore the properties, structure, types, and functions of RNA. So, let's get started. Now first, what is RNA? RNA is a polymer made up of nucleotides, which are the building blocks of this molecule. Each nucleotide consists of three essential components: a ribose sugar, a phosphate group, and one of four nitrogenous bases. RNA is known as ribonucleic acid because of this ribose sugar. It's slightly different from the sugar found in DNA, as it contains an extra oxygen atom. The structure and function of RNA are significantly affected by this small modification. Moving on to the nitrogenous bases, RNA shares three of the bases found in DNA: adenine or A, guanine or G, and cytosine or C. However, instead of thymine, RNA has uracil or U as its fourth base. Uracil can pair with adenine, forming hydrogen bonds, just like thymine does in DNA. The unique combination of a ribose sugar and these four nitrogenous bases allows RNA to perform a wide range of essential biological functions within cells. RNA is involved in various biological roles, including coding, decoding, regulation, and expression of genes. It acts as a messenger molecule that carries the genetic information from DNA to the cellular machinery responsible for protein synthesis. Let's take a closer look at the structure of RNA. Like DNA, RNA is a long polymer made up of nucleotides. However, some important differences set RNA apart. First, RNA is a single-stranded helix, unlike DNA's double-stranded structure. This single-stranded nature gives RNA unique properties and functions. The RNA strand has a 5' end with a phosphate group and a 3' end with a hydroxyl group. These ends play crucial roles in the synthesis and function of RNA molecules. Now, let's talk about the building blocks of RNA. As mentioned before, RNA is composed of ribonucleotides, which are similar to the nucleotides found in DNA, but with some notable differences. The ribonucleotides in RNA consist of a nitrogenous base, a ribose sugar, and a phosphate group. The nitrogenous bases in RNA are adenine, cytosine, guanine, and uracil. Interestingly, uracil replaces thymine, which is found in DNA. However, uracil can still pair with adenine, just like thymine does. Another difference is in the sugar component. In RNA, the sugar is ribose, whereas, in DNA, it's deoxyribose. This distinction in sugar type gives RNA its unique properties and allows it to perform various functions in the cell. While most RNA molecules are single-stranded, certain regions within an RNA molecule can fold back on themselves and form complementary base pairings. This results in the formation of double-stranded regions, creating what we call a secondary structure. Examples of RNA molecules with substantial secondary structures include ribosomal RNAs, or rRNAs, transfer RNAs, or tRNAs, and even some messenger RNAs, or mRNAs. This secondary structure is essential for their proper functioning within the cell. Now look at the types of RNA. There are three main types of RNA found in both prokaryotes and eukaryotes. Let's explore each of them. First one is messenger RNA, or mRNA. Messenger RNA, or mRNA, is a type of RNA synthesized in the cell nucleus and then transported out of the cell. mRNA is translated into polypeptides, which are the building blocks of proteins. The size of mRNA reflects the size of the polypeptide it encodes. Most cells produce thousands of different mRNA molecules in small amounts. Many mRNA molecules are common to most cells and encode proteins that guard cell metabolism. Some types of mRNA are specific for certain types of cells and encode proteins that are needed for the function of that particular cell. A mature mRNA of the eukaryotic cell is made up of 5 subunits, which are the 5' cap, the 5' untranslated region, or UTR, the coding region, the 3' UTR, and the poly-A tail. The 5' cap is a modified guanine nucleotide that is added to the 5' end of the mRNA. It protects the mRNA from degradation and helps to initiate translation. The 5' UTR is a region of non-coding RNA that is located upstream of the coding region. It can play a role in regulating translation, stability, and export of the mRNA. The coding region is the region of the mRNA that contains the genetic information for a protein. It is translated into a protein by a ribosome. The 3' UTR is a region of non-coding RNA that is located downstream of the coding region. It can play a role in regulating translation, stability, and export of the mRNA. The poly-A tail is a region of polyadenylate, or adenine nucleotides that is added to the 3' end of the mRNA. It protects the mRNA from degradation and helps to export the mRNA from the nucleus. Second is ribosomal RNA, or rRNA. Ribosomal RNA, or rRNA, is a type of RNA that is found in ribosomes. Ribosomes are the organelles that make proteins. rRNA makes up about 60% of the weight of a ribosome. rRNA has a distinctive three-dimensional shape that helps it bind to mRNA and tRNA and catalyze the formation of peptide bonds between amino acids. rRNA also helps to determine the shape of the ribosome. The proteins on the ribosome surface stabilize its structure by interacting with the rRNA core. Ribosomal RNA, or rRNA, is a type of RNA that is essential for protein synthesis. rRNA is synthesized in the nucleoli of the cell nucleus. Prokaryotic ribosomes are made up of a small subunit, which is 30S, and a large subunit, which is 50S. Eukaryotic ribosomes are made up of a small subunit, which is 40S, and a large subunit, which is 60S. Ribosomes are associated with the endoplasmic reticulum or can be free-floating in the cell cytoplasm. And third one is transfer RNA, or tRNA. Transfer RNA, or tRNA, is a type of RNA that carries amino acids to the ribosomes. It acts as an intermediate between nucleotide and amino acid sequences. tRNA molecules form hydrogen bonds with mRNA and ester links with amino acids. This allows tRNA to bring the correct amino acid to the ribosome for protein synthesis. tRNA, or transfer RNA, is a small molecule that helps to build proteins during translation. It carries specific amino acids to the ribosomes based on the instructions from mRNA. tRNA has an anticodon that pairs with mRNA, allowing the correct amino acid to be added to the growing protein chain. It has a unique structure with regions called the D-arm, T-arm, and anticodon loop. These parts stabilize the tRNA and interact with ribosomes during translation. The tRNA also has a sugar phosphate backbone and specific ends called the 5' and 3' ends. Overall, tRNA plays a crucial role in protein synthesis by delivering amino acids to the ribosomes. Apart from these three main types, there are also other important RNA molecules. Let's briefly discuss them. First, small nuclear RNA, or snRNA. Small nuclear RNA, or snRNA, are short RNA molecules that play a role in the processing of pre-messenger RNA, or hnRNA, in the nucleus. They are transcribed by RNA polymerase II, or RNA polymerase III, and are about 150 nucleotides long. snRNA has different genes in multiple copies, which play different roles in the synthesis of other RNA classes. For example, snRNA is part of the spliceosomes that help in the conversion of hnRNA into mRNA by excising the introns and splicing the exons. They also mediate the regulation of transcription factors and RNA polymerase II. Additionally, snRNA maintains telomeres. snRNA associates with specific proteins and complexes, known as small nuclear ribonucleoproteins, or snRNPs. Second, small nuclear RNA, or snoRNA. Small nuclear RNAs, or snoRNAs, are small RNA molecules of about 60 to 300 nucleotides, found in the cell nucleolus. They play a role in the synthesis of ribosomes, the modification of nucleotides in rRNA, tRNA, and snRNA molecules, and the splicing of pre-mRNA into different forms of mature mRNA. One type of snoRNA serves as a template for the synthesis of telomeres. In vertebrates, snoRNAs are made from introns that are removed during transcription. Third, microRNA, or miRNA. MicroRNAs, or miRNAs, are small, single-stranded RNA molecules that play a role in gene regulation. They are found in plants, animals, and some viruses. miRNAs are encoded in the genome and are expressed in some cell types at certain times during the differentiation of these cell types. miRNAs regulate gene expression by either destroying the mRNA or repressing its translation. miRNAs are small and do not need to be translated into proteins in order to regulate gene expression. Genomic studies of mammalian gene expression have revealed that one or more miRNAs can bind to an mRNA that has been transcribed from DNA. A single miRNA can bind to about 200 different mRNA targets, which allows coordinated mRNA translation. Fourth, long non-coding RNA, or lncRNA. Long non-coding RNAs, or lncRNAs, are a large group of RNA molecules that are not translated into proteins. They are about 200 nucleotides long and are encoded in the human genome. lncRNAs are involved in a variety of cellular processes, including gene regulation, splicing, translation, and imprinting. One of the most well-known lncRNAs is xsTRNA, which is involved in the inactivation of one of the two X chromosomes in female vertebrates. xsTRNA binds to the X chromosome and recruits proteins that silence the genes on that chromosome. This process is called X chromosome inactivation and is essential for maintaining a balance of gene expression between males and females. lncRNAs are also involved in the regulation of gene transcription. They can bind to enhancer and promoter regions of genes and help bring these regions together. This process is called looping, and it helps regulate the expression of genes. Okay, now look at the functions of RNA. RNA serves various essential functions in the cell. Let's explore some of its key roles. 1. Protein synthesis. Protein synthesis mainly depends on RNA, where mRNA carries the genetic information from DNA to the ribosomes. rRNA helps in protein synthesis at the ribosome, and tRNA delivers amino acids to the growing polypeptide chain. 2. Gene regulation. Certain types of RNA, such as miRNA and snRNA, play significant roles in gene regulation, controlling when and how genes are expressed. 3. Enzymatic activity. Some RNA molecules, called ribozymes, exhibit enzymatic activity and can catalyze biochemical reactions within the cell. Remember, besides these very important functions, there are more other functions of RNA. And now there you have it, the properties, structure, types, and functions of RNA. The fundamental functions of life are greatly impacted by this amazing molecule. Remember, RNA is not just a simple messenger, but a versatile player in cellular activities. Thank you for watching. Keep exploring the wonders of biology, and for more exciting topics, please don't forget to subscribe to The Whiteboard Biologist. See you in the next video.